All right. Hello, everybody. My name is Hugh Simmons, and today I'm going to take you through my research review into Trypanosoma brucei, specifically focusing on the infection cycle of the parasite. A little bit of background. It is a parasitic protozoan endemic to Africa. We read about it. Is the pathological cause of African sleeping sickness or human African trypanosomiasis, also animal African trypanosomiasis. Of note, it does exist in 3 subspecies. We're going to be focusing primarily on TB gambiense and TB rhodesiense today. As mentioned, the infection cycle exists in 2 stages. We have the mammalian, or more like the reservoir and pathological infectious stage, as well as the tsetse fly stage, which acts as the vector of transmission and infection. We're going to be staying over on the right side of this in the mammalian stages, focusing a lot on early infection, animal reservoirs, and how we can best prevent the parasite from getting to humans in the first place. The first article is from the peer-reviewed journal Parasite, titled Molecular Identification of Trypanosoma brucei gambiense in Naturally Infected Pigs, Dogs, and Small Ruminants Confirms Domestic Animals as Potential Reservoirs for Sleeping Sickness in Chad. The goal, as you can read there, was to identify potential reservoirs in domestic animals that can serve as vectors of transmission. They propose that these reservoirs do play a crucial role in the endemic nature of the disease, preventing its complete eradication. The findings, as you would expect, are that as animals and humans are living in close proximity to each other, and those animals are found to have cases of the disease as animal reservoirs, the humans are generally more likely to contract the disease. So this is a study point that seems like common sense, but also having the data to back it up is very important. This provides a launching off point for further investigation into the control of animal infection to help further prevent human infection. The second article from Nature Communications is Vector-borne Trypanosoma brucei parasites develop in artificial human skin and persist as skin tissue forms. Originally, this study was done to find a reliable artificial substitute for human tissue to be able to study the early infection cycle of T. brucei in a laboratory setting, in a controlled setting. They were able to do that. They came up with a high-density skin equivalent that, using tsetse flies that they knew were positive as a vector for the disease, they were able to infect the skin through a natural tsetse fly bite. Of note from this, they had an unexpected finding of the actual parasite existing in 2 separate forms within the human host. Within the skin itself, it exists as the skin tissue form prior to infecting the bloodstream, which is the pathologic cause what we find the patients actually undergoing African sleeping sickness. The bloodstream form is what is deadly. The skin tissue form, however, is a very low metabolic state in which the patients are asymptomatic. So at this point, the human host can potentially act as a reservoir within the human settlement or village. So we have the animals living domesticated, as well as humans who are asymptomatic, potentially as a hidden reservoir for what could essentially appear as sporadic disease development within a village that otherwise appears to be healthy. Third article from Microorganisms is the current treatments to control African trypanosomiasis and One Health perspective. This took a look at the current practices and trying to come up with suggestions for best practices for the future. Currently, the practices are fallen a little short. Like, there has been efforts and the disease has been on the decline, yes, but to be able to actually eradicate the disease with a goal of 2030, there's overall poor research investment and lack of communication or networked efforts with this. There are multiple reasons, one of which is as the disease decreases and becomes harder to find, the incremental cost is higher, and there is a lack of potential revenue for private investment from pharmaceuticals on the back end. They came up with 6 points, essentially better networked, better communication, and better coordinated efforts. Effective surveillance infection among wildlife, domestic animals, and humans. Better studies into the vector surveillance and control measures. We'll talk a little bit about that on the next article. Communication systems to allow effective notification of identified infectious cases. A development of effective, sensitive, and fast field diagnostics. That also is touched on with the next article. networked communications between stakeholders, medical and veterinary departments, and governmental policymakers to essentially better spread the word of when, where, and how the infections are happening, and safer and simpler treatment paradigms. Currently, the treatments that are available have large side effects, major side effects that make sticking to the treatment difficult. And as we know, if you don't go through a full, proper antibiotic or antimicrobial treatment, then all we're doing is causing or aiding in the evolutionary development of resistance to the treatments that are available. The final one is a pilot study demonstrating the identification of T. brucei gambiense and TB rhodesiense in vectors using a multiplexed high-resolution MELT qPCR. This is touching on trying to find a simpler, more efficient, cost-effective detection screen to identify areas of risk, focusing on the tsetse fly rather than post-human infection. With their advanced screening techniques, they had a higher sensitivity, lower detection threshold, and faster time to resolution with more objectively analyzable results rather than subjective reading of the results. The single assay can test for both TB gambiense and TB rhodesiense, and testing tsetse flies allows for the identification of areas of concern prior to confirmed human infection. So focusing on, all right, we're seeing an increase in the tsetse fly carriers of this, we can focus prophylactically on the prevention of human infection rather than waiting for the infection and outbreak in humans. Finally, the ongoing fight for T. brucei, noted from these articles, is difficult. There are many factors that go into it: socioeconomic, environmental, evolutionary, and communication between governmental and private sectors. The close proximity of the animal reservoirs that are known to the humans, as well as hidden human reservoirs as asymptomatic carriers, causes difficulty in complete eradication. The cost is going up and up to find the disease as it becomes more rare, and the efficiency becomes paramount. The cost effectiveness to make it worthwhile for us to be able to garner more money into the research and get more eyes, hands, and knowledgeable minds involved. Finally, these are my references. I have the 4 articles as well as the CDC infection cycle graphic. I know I learned a lot about T. brucei, both gambiense and rhodesiense, and the infection cycle, as well as African sleeping sickness. I hope you learned a little bit about it today. I know it was really fast, but trying to get it as short and sweet as possible for you. Everybody have a great day.